Welcome to in-person genetics colloquium. Yes. Today we have a very exciting speaker, Chad Myers, who came to us all the way from Minnesota in his car. So he wanted it, and that makes sense because this is a fantastic audience to present to, and we're going to look forward to a fantastic talk from Chad. So I don't want to go on too much, but you might be wondering why a bacteria dude like myself invited someone who has a top title that says human genomes. And I think what it really shows is the impact that Chad has had across all fields of biology in functional genomics. He's really come up with the quantitative underpinnings for genetic interactions, chemical gene interactions, and establishing gene networks. So his impact has been very broad. You can feel it in the microbial community and in the human genetics community. So I'm very, very excited. I hope you're excited. Here's Chad.
Thanks a lot for that nice introduction and for the invitation, Jason. I also continue to be part of the microbial community. So I might talk about humans today, but I'll talk about yeast tomorrow sometime. So I'm happy to be here in person. This is my first in-person seminar for a while. So this is a reappreciating things about life again. So I want to encourage, I'm happy to not get through these slides. I'd rather have people interrupt me during the talk. Let's take advantage of the fact that it's in person for some of you. So feel free to stop if you have questions during the talk. I'd rather have a discussion, even if it means I don't get through the slides that I have. So I'm going to talk today. So my lab is broadly interested in genetic interactions, has been basically since I started it. We've worked on genetic interactions from yeast all the way up to human genomes. I'm going to try to give you a narrative today that starts in some of our yeast work and talks about some of our efforts to translate that to try and understand genetic interactions underlying complex disease. So that's a kind of preview for the talk. So just context for the talk. So I put together this video a while ago, which captures what we've learned about human disease or trait associations over the last couple of decades using GWAS, genome-wide association studies. So I think most of you are familiar with genome-wide association studies, genotype populations of individuals at many points across the genome, let's say 500,000 or 1 million different points in the genome, collect a set of cases and a set of controls that focus on a specific disease, and then look for one-by-one associations between a single nucleotide polymorphism in the genome and the trait of interest. And this approach has been highly productive over the last couple of decades. This video kind of captures that. This shows, as studies were published, how it filled in knowledge of associated loci with different traits. So each one, this is the human genome map. Each one of those dots is a different association that's been mapped for a given trait. And I just checked the numbers again today. There's something like 347,000 SNP trait associations across something like 5,600 different studies. So the approach has been applied over and over. It has been highly successful, and that map is a lot fuller than it was back in 2006. And we've learned a lot about individual loci associated with human disease. So that's a kind of zoomed out view of that, about that same picture. So one issue with this, and this has been described in a lot of different ways, the concept of missing heritability is a bit controversial. I'll preface what I say with that, but the problem still remains. So while this has been highly successful at identifying loci that are associated, if we turn around and try to predict disease risk from loci that we know about, for pretty much any disease, most human traits with a few exceptions, there's a gap between what we should be able to predict and what we can actually predict from that loci. And that's been referred to by a number of people as missing heritability. There's a lot of reviews on this. As I said, it's a bit controversial what exactly constitutes missing heritability, but I take a fairly simple view of this. Basically, we can measure what fraction of disease should be genetic, which fraction of that risk should be inherited, passed down from generation to generation. That varies by disease. And then we can take the fraction of that and ask how much of that can we explain. Let's say if I build a model that just uses the loci that we know about to predict disease risk. And for most diseases, the proportion of heritability explained that we should be able to explain is quite low. So these numbers are slightly out of date for some of these traits now, but it ranges from say 50% on a good day or for a good trait that we actually understand quite well, down to say 1 or 2 or maybe 5% for many diseases. So there's a big gap between diseases we know are genetic and our ability to explain those with the loci we know about. And I should say height, I think, is an interesting exception to that. I think height, if you look at the numbers published today, we're explaining way more than half of the variation, I think, with loci that we know about. And that's due to much larger cohorts, we've been able to find a broader set of associations. So there's a lot of explanations for why this might be true. And I think it's important. I'm focusing on one area today that is one possible explanation, and likely all of these contribute in some form. But it could be that many low effect single associations explain this. So maybe as we get bigger cohorts, and we have cohorts like the UK Biobank these days with several 100,000 individuals, we can discover variants that we couldn't previously detect before. This fills in some of our knowledge of what puts people at risk for disease. Another explanation that has been explored in many disease scenarios, our work hasn't addressed this very much, but rare variants. So genome-wide association studies almost exclusively for the first decade or so focused on 5% variants of 5% frequency or more. So if variants in the population that are rarer than that contribute to disease, these approaches are largely going to miss them. And there have been a lot of studies that indicate that that is the case in some diseases, that we've just missed really strong effect, but rare alleles in populations that cause disease. The explanation that I have been working on for several years now is that maybe complex disease, a complex genetic basis is behind this missing heritability. So all of the models that we use to explain heritability, for the most part, assume kind of an additive contribution of independent loci contributing to that disease. That does not allow a strong contribution of specific combinations of loci that might have a strong effect. And there's a good reason why we focused on the single variant approach, why that's been our paradigm. It's because it's a very challenging problem to try to tackle the combination of variants, to take a combination of variants approach. So the current paradigm basically is to step through the genome looking at variants like this. So a single nucleotide polymorphism A, say, test that for individual association with the disease, go on to the next one, test that for individual association with disease. Already there's a big multiple testing issue there. So for a standard genotype, you might test 500,000 or 1 million different locations in the genome. Of course, some of those are linked to each other, but the number of tests is still quite large, even for single variants. So the pairwise version of that problem looks like this. Let's go through, let's say 500,000 or 1 million different loci and take all possible pairs of those. Let's say that what allele does a given individual have at this location A? What allele do they have at location X? Let's group individuals that have the same combinatorial, the same joint genotype in both of those, and then let's do an association with those. So the multiple testing problem I described for single variants is a whole lot worse for that combination of variants case. Even if I restrict my set to 500,000 variants, say, that's something like 500 billion tests. And for the most part, cohorts that we look at these days just can't support that number of tests and still come out with statistically significant results. The larger cohorts, UK Biobank, if I get into the hundreds of thousands of individuals and I have a really strong effect size, I might be able to approach that problem. But for the most part, we can't detect, unless these have a very strong effect, using this single pairwise variant approach. So that's the problem with trying to find genetic interaction. So I'm gonna claim today that we have a way to approach that problem. We're gonna change the question a little bit, but I think still find interesting combinations of variants that associate with disease. And I'll come back to that human problem in just a second. I'm gonna take a little detour through my favorite model organism, Saccharomyces cerevisiae. So I've worked on yeast really since I was a graduate student, and there's a lot of power in things we can do with yeast, as many of you here can attest. So we've learned a lot about genetic interactions from this simple model yeast as well. And I'm going to focus on a very simple phenotype from yeast. Basically, it's how fast yeast colonies can grow in the absence of specific single or combinations of genes. And that's all basically phenotyped on plates. This is an agar plate about that big with a collection of mutants that have been grown up over a couple of days. And so that simple phenotype, basically fitness in the absence of single or combinations of genes, we've learned a lot about genetic interactions. And maybe just to motivate this, I think for some of you it's obvious, but if we hope to understand genetic interactions, this provides a much cleaner platform for doing that. I can take a single genome, make 2 precise mutations, and only those 2 mutations make the corresponding single mutations and quantify the effect of those. If I can't understand genetic interactions in that context, there's no way I can understand them in the more complex human context. So all of our work in genetic interactions in yeast is with a couple of very close collaborators, close friends of mine, Charlie Boone and Brenda Andrews, who are at the University of Toronto. Over the last, I guess, more than a decade now, we have been working together. They really pioneered the experimental side of this. So I had very little to do with setting up the experimental side of this yeast genetics platform, but they pioneered an approach called synthetic genetic array analysis. Basically, it is a robotic way of constructing double mutants very efficiently from well-defined mutant collections. So they can construct mutants, double mutants precisely and rapidly in phenotype with these auger plates that I just mentioned. So that's a picture from their lab that shows some of the robots they use to do that. So just to emphasize what we're quantifying is basically we're quantifying the area of these colonies. So that's a proxy for fitness over a few generations that we've grown them up on these plates. So we've worked with Charlie and Brenda's labs over the last several years to quantify things reliably from these array-based platforms. I'm not going to go into the details behind that. I've talked with the students a lot at lunch. I think 80 to 90% of what computational biologists do, and I think one of the reasons why they're important is we're taking raw data into things that are actually interpretable. And there's a lot of work that happens in that arrow right there, from raw image data to data that's worth interpreting because you've removed all the systematic variation. I'm not going to talk a lot about those details, but that's not to say it's not important. But a lot of that's published. I'm happy to talk offline if people are interested in some of the statistical challenges of quantifying genetic interactions from that kind of phenotype data. At a high level, what we do is quantify single and double mutant phenotypes from these mutants. So basically, we measure quantitative fitness phenotypes for single mutants. Let's assume this mutant A, in this case, we get a 0.7 fitness. That means in one wild type generation, the A mutant lacking, or the mutant lacking gene A constructs 70% of a cell. So it's basically a measure of fitness. Let's say a mutant B, I get an 80% fitness phenotype. A null model that is often used in the context of fitness phenotypes, when these genes have nothing to do with each other, I expect those to combine multiplicatively. So I expect to get a 0.56 fitness in a double mutant that combines both the A and the B mutation. And we're looking for cases that differ interestingly from that. So I'm looking for cases where the double mutant is a lot sicker than we expect. So let's say it's 0. That would be it's dead. That would be a case we call synthetic lethality. There's also negative epistasis. I'll use the word negative and positive for these signs. Positive, on the other hand, is a case where the double mutant is much healthier than I expect, and that's something that we can that tells us something different about the relationship between those genes. So, as I said, there's a lot of details on how we can quantify these numbers from arrays. Just to summarize that, basically we can construct really millions of double mutants, measure single mutant phenotypes, double mutant phenotypes, and measure a quantitative number that reflects that interaction that I just described. So that's the sort of quantitative phenotyping that's been done in yeast over the last couple of decades. And Charlie and Brenda's labs have been turning the crank on this approach for many, many years. So to date in yeast, in the single sort of standard laboratory conditions, they've constructed something like 23 million unique double mutants. It's about 90% of what's possible. Based on the reagents we have right now, there are some details about reasons we can't construct some of the others. So pretty exhaustively cover the space of double mutants that are possible, at least in a single laboratory condition. So I'm not going to go into tons of detail on the yeast results other than to say about 1 million of those cases showed phenotypes that are different than what you expect. They don't fit the null model. About 550,000 are negative interactions, that they're sicker than we expect. And something like 350,000 of those are positive interactions at the threshold that we're reasonably confident in calling those interactions. So a lot of that work is described in the 2016 paper, where that was our last installment of the main yeast genetic interaction mapping effort. I should say, I'm not going to talk about it today, but one of the other ongoing efforts has been to map double mutant interactions across a variety of different conditions. So as I just described, this is done under a single lab condition. The answer, or the question we always get is, well, what happens if I change the conditions, if I remove this element of the media, put yeast under some stress, add this drug? And of course, the network changes, and that's interesting to study. We've just published a separate study just last year where we did the same experiment on a smaller scale, but across 15 or 16 different other conditions that stress out the cell in various ways. What I will tell you from that is that the network is surprisingly stable. So I think previous studies that have studied smaller sets of genes, not surveyed such a systematic set of conditions, reported dramatic changes. There are changes in that network, but the core of it is quite stable. So I would say we refined a lot of the same interactions, and we estimated after screening across 15 or so conditions, it's very difficult to find additional new interactions without very specific stresses. So generally, we think this is a good reference map that we can use to understand a lot of the basic biology of yeast. Let me pause for a second. Are there any questions that people have so far? I'm happy to stop and talk about specific things. Okay. So that's basically what we got from these, and this summarizes the quantity. This is sort of a quantitative summary of that. So maybe just to emphasize a little bit the quantitative model I talked about. So if I measure the difference between what I observe and what I expect based on this multiplicative model, this is just a histogram of this epsilon value that I'm describing here. So what you can see is that the vast majority of pairs fit that null model. That is, there's a peak at 0. This is just a log scale version, a log scale of the y-axis, just to show you that there is actually tails here that you can see if you blow that up a little bit. So everything that's colored in red here are cases that we can confidently distinguish from 0, and things that were colored from green are confidently distinguished from 0 on the positive side. The bottom line is it's about 3% dense in terms of negative interactions and 2% dense in terms of positive interactions. So it's relatively sparse, but I think one important takeaway from this is, think about it like yeast has about 1,000 or 1,100 essential genes. That is, genes that when you knock them out, you can essentially completely kill the yeast cell. There's something like 10 times that, maybe 20 times that. If I only look at the really strong effects, that is mostly dead in the double mutant, there's about 10 times the number of cases where if I'm allowed to take 2 perturbations, I can kill the yeast cell. So there's a lot of ways using complex genetics that we can arrive at pretty extreme phenotypes. And that's something that motivates our searches for these in the case of human disease. We think that while they're rare, they could overall explain a number of fairly severe or extreme phenotypes. I should also mention they're rare, but they're several-fold more dense than physical networks. So PPIs, protein-protein interactions, have been mapped extensively in yeast. The biggest versions of those networks are on the order of tens of thousands of pairs. So genetic interaction networks tend to be much more complex, just denser than physical networks. With that complexity means complexity of interpretation as well. So they're harder to interpret. It's not a physical connection between genes. This is something that makes them more difficult to interpret. So let me also just mention something else that we're doing a lot with genetic interaction that we and other people have done. I think maybe the most powerful thing about genetic interaction networks are that they're an unbiased way to understand how everything's related in a given genome. So this has been a paradigm that's been used over and over again in our field. But basically, if I take a matrix of interactions, let's say I've taken each one of these genes and crossed it, constructed double mutants with each of these genes, and the colors here indicate a sign of interaction. Really, we have a quantitative network here. One of the simplest things you can do, and also the most powerful things you can do, is to simply cluster these networks such that I look for genes that have very similar profiles. So gene A here has the same positive interaction partners and negative interaction partners as gene B. That is, they cluster near each other here. They have high correlation. That typically indicates that those two genes well, they phenocopy each other in this case, which means that the phenotypes I get when I combine mutations in A with mutations in other genes mimic what happens when I do the same for gene B. So, and this often gets you to very specific local relationships. I can pinpoint members of the same protein complex just by looking for things that are highly correlated in these networks. So we often take a direct interaction network and transform it into a profile similarity network because it allows us to directly interpret those interactions. And that's what that looks like on a genome-wide scale in yeast. If I do that, so the nodes here are all the genes in yeast. The edges are now profile similarity, so the genes are connected when they have a highly similar profile to another gene. And this is just a spring model layout based on Cytoscape. But what you can see there are nice structures that form, natural structures that form all over the place. In yeast, we know a lot about genome annotation over the last couple of decades. The yeast community has done a great job of tracking and both figuring out biology and also tracking what we know about yeast biology. And if you overlay known functional annotations on top of that, you can find enrichment for very coherent neighborhoods all over that map. So what this says is if you can take a cell, take a genome, perturb it in very precise ways in combination, measure a very crude phenotype. Remember, I'm not doing microscopy. We're not measuring specific reporters. We're basically just taking a snapshot of what yeast colonies look like after a couple of days. Really, we can learn about biology in a very specific way all over the map. A lot of processes cluster very well on that network, which I think, to me, is very promising, exciting. This is why we're working hard to push these same approaches into human cells. It's an unbiased way of us figuring out how all the genes are related. What are the pathways that they're all connected through complexes? What are the functional modules that they participate in? So if you take nothing else away from my talk about genetic interactions, I think this is one of the powerful things that they can give us, that regardless of how crude the phenotype is, we can do some pretty powerful things. And I should say, I'm not going to dwell on this, but we have used that. Charlie and Brenda, a bunch of our yeast collaborators, have gone in and specifically characterized new genes, functions based on where they fall on this map. Even in yeast, there's still genes left to be characterized. Surprisingly, a couple of years ago, there was even essential genes that were uncharacterized. So these types of approaches can really shed light on very specific hypotheses about where these genes function based on where they fall in that map. So that's a little bit about sort of at a high level what we learned from yeast. I want to zoom in on a very specific observation that motivates all of what we're doing in human cells and in human populations, I should say. One of the really striking observations from yeast, and again, this is not just our work, but Nevin Krogan, Trey Eidecker, a bunch of other people in the yeast space, in the yeast genetic interaction world, have observed very similar things. These networks are highly structured. So what I'm showing you here are a couple different protein complexes. So SWARE1 complex and RPD3 complex. What you see here are negative interactions in red and positive interactions in green. Each one of these is an individual gene coding for a protein in those complexes. And remember, these networks in general are sparse. So there's something like 3% edges across the whole thing. But if I zoom in on these little subnetworks, you see a different story. So basically, almost every single pair between this set of genes and this set of genes, these are two separate complexes, is connected with a negative interaction. The same thing if I focus, this is the 19S proteasome, so the proteasome complex and a few other complexes around the outside. Same thing, if I find a pair of complexes that interact, the density of interactions crossing those two complexes between any pair of genes between those complexes is very high. So once I get into a space where there's interactions, you find these bridge-like connections all over the place. These are, in computer science terms, we call these complete bipartite graphs. So you have a set on this side, a set on this side, and they're completely connected across them, which is highly non-random structure in such a sparse network. So we've quantified how much that structure characterizes the yeast network, and we've exhaustively discovered. So there's a whole field of association rule mining from computer science that actually applies quite well to this problem. We can take the yeast network, and we don't have to know what the pathways are in advance. We don't have to know what the complexes are in advance, the protein complexes, and we can just let the network tell us what these structures are. And we've done that. So in yeast, we've basically characterized all the the higher level structures that there are. And what we can tell you is that there's something like 75% of interactions you find in yeast are embedded in one of these larger structures. So if a yeast biologist comes and tells you, I knocked out these 2 genes and it killed the yeast cell, 75% of the time, that's not a local observation. That is an observation that reflects something about the pathways that those are involved in. And the structures that we looked at here are on the order of 3 genes or larger. So these are generally large structures, and some of them go up to 5, 10 genes at a time. So these blocks of interactions characterize the yeast network. Some of them I haven't distinguished so far between this one on the right and this one. This reflects a structure where I'm getting interactions within the same set of genes. We call these a within pathway or within protein complex interaction. That does happen as well. These are much more frequent. There's many more possibilities for these between pathway interactions. But both of these happen. And together they explain about 75% of the negative, the synthetic lethal, synthetic sick type interactions in the network. So that's the major jumping off point for what we wanted to translate to the human genetics question. So I'm going to jump back to human genetics in a second here, but I can pause if there are any questions if people have them at this point. That's the key observation that motivated everything that we've been doing recently in the human genome. Yeah, there's a couple of questions back here. Go ahead.
Okay. So I saw that in your kind of like circle with the different types of genes around it, you did have a circle that talked about like non-coding DNA. And I'm interested in, because I know that you're talking about like interactions between genes and like genetic pathways, but I'm interested in what your method would be able to tell us about the role that like non-coding DNA might play in those pathways.
Yeah, it's a great question. I would say in yeast, we haven't done anything on that front, and yeast has fewer, well, there's interesting non-coding, there's ribosomal RNAs, things like that play a hugely important role. I think it's a great question. We have not cracked open that question. I think in human cells, for instance, with CRISPR approaches these days, it's quite easy, and there's libraries designed to target those non-coding elements. I think there's been some limited studies that have looked at phenotypes for non-coding elements. We haven't done that yet in the interaction space. It's a great frontier to explore, and there's going to be interesting things that we can find from that, especially if you're using a CRISPR approach to target. There's no reason we can't target those elements and figure that out. In yeast, we have not addressed that non-coding space. I think there was another question up front. Yeah.
I was wondering, are there any genes that you've encountered that you've deleted that don't interact with anything else you tested?
Yeah, that's a great question. We call this the dark net. And yeah, I would say, in yeast there's 6,000 genes. About 2,000 to 3,000 of them really dominate that network. I should also say I didn't talk about this here, but we've characterized what are the properties of genes that do have a lot of interactions. Probably the most striking property is they have single mutant phenotypes that are detectable. So generally, genes that have very subtle, it might even be a 3% fitness defect as a single mutant. Those are the sweet spots to go to look for genetic interactions, because that's where we tend to find them. There are a whole bunch of other genes that are pretty silent in the network, I would say. They don't connect well to that big network that I showed you. And disappointingly, we always thought, okay, we're going to go after other conditions and we're going to connect all those things in. That didn't happen. I would say the dark net continued to be the dark net even after we stacked 15 other conditions on top of it. There are certain genes that are maybe due to redundancy, genetic redundancy. There's a lot of larger families. Also, maybe we just haven't hit the right niche condition that gene is really required under. We can get a phenotype. We're not looking at the right phenotype, right? So we're looking at a very crude phenotype. If we look at the right phenotype, I'm confident we could find a place where those genes have a phenotype likely. But there's a decent chunk of the network that just isn't well connected, and I would say we haven't shed a significant light on that with extra conditions either, unfortunately. Any other questions at this point? Good question. So, okay, so I want to transition. So that was yeast. I want to transition back to the human question. So we worked in yeast. We continue to work in yeast. We have a number of projects going on looking at interactions in different genetic backgrounds, different conditions. But we wanted to take, several years ago, we wanted to take some of the insights we had about genetic interactions from our yeast work back to the human complex disease question. So can we develop better methods, better computational methods for finding genetic interactions from GWAS-like cohorts if we leverage some of what we know from yeast? And the idea is pretty simple. This is a standard GWAS. We have a method called bridge. So bridge takes in a standard GWAS-like cohort. So you need about 1,000 disease control individuals. Obviously, the more the better. The more we have, the more power we have to detect these things. But it can work on cohorts as small as 1,000 or a couple thousand individuals. Another key input to this approach is you need to tell us what the sets of related genes are. So what we're going to do is use the fact that that these interactions are structured in order to reduce the number of tests we do and group individuals that might have totally different variants, different combinations of variants, treat them as similar because they affect similarly functioning genes. So the input, basically genotype data from humans that are in this case control or a quantitative phenotype is also fine. A collection of gene sets, so we can derive these from established curated pathways. We're doing more work these days on deriving these from unbiased integration of genomic data, but these are sets of genes. They don't necessarily need to be related to the disease, basically collections of genes that I know are functionally related. And our approach takes these and looks for structures that looks like this. So the approach, I'm just going to describe this at a high level, and I can go into more details if people want. But basically what our idea was, rather than look for an individual interaction between two SNPs, let's say this SNP B and SNP X, and we already talked about how challenging it is to tackle that problem. That is, test all possible pairs that are like B and X, because it's half trillion tests if I just look at all combinations of SNPs in a standard GWAS. I'm not going to have the statistical power to do that. But what we said is, if you instead step back and say, if you find interactions that are likely structured, let's assume for a second that interactions are structured like in yeast. That is, they follow this between pathway model like we discovered in yeast. When you find one interaction crossing two genes in the same pathway, chances are there's probably going to be other potential for interactions between other pairs of genes crossing the same bridge, the same two pathways. So that's the simple idea that we took from yeast. We're not assuming conservation at all. So the idea is not to map interactions that are conserved from yeast up to human. I think that's another interesting direction. Some people have worked on that. All I'm saying is assume this prior on the structure that these bridge-like interactions should exist, and let's change our statistical test rather than testing for individual pairs, instead to test for these bridge-like structures in the human data set. And these are what we call between pathway interactions. I can extend the same idea to within pathway interactions, and also test at the sort of single pathway level for that interaction. So this summarized that process more in words. Basically, we map SNPs to established genes and the corresponding gene sets. So as most of you know that have worked in this space, there's a lot of variance to solving that problem. How do you map genes or how do you map SNPs to the genes they're associated with beyond genetic distance? There's a lot of interesting, we can use some of the ENCODE type data to make those associations these days. What we do basically is construct a network of individual pairwise SNP interactions. So actually we go through all possible combinations of SNP level interactions, test for association with the phenotype. So I construct what we consider a very noisy SNP level network. And then we step back and we look for pathways that have an abundance of these SNP level interactions crossing them. So our hypothesis test went from single SNP pair to a pathway pair, and we're looking for an abundance of edges crossing those two pathways with SNPs links to genes in those pathways. And of course, we have to estimate the significance of anything we find there from a statistical standpoint. And we do that both through SNP permutations as well as sample permutations. We permute the sample labels, and we can assess how significant is any of these structures that we find. So at a high level, that's the bridge approach. There's obviously a lot of details there. I haven't told you anything about disease model assumptions. So we can use an additive disease model. We can use a dominant disease model where we assume that if an individual has a single occurrence of a minor allele in two different loci, that might predispose them to the disease. We can use a recessive model. So there's a couple different switches that you can dial here in terms of this algorithm. I'm going to skip some of those details. I'm happy to talk offline, but that's the core of the idea. So just to give you a sense for what an individual discovery looks like. So we've worked a lot in the case of Parkinson's disease. This is an example of an interaction that you can discover with this approach. So this is a very complex slide, but I'll walk you through these details. So basically what each one of these bars here reflects an individual SNP. So these are, if you're doing a genome-wide association test, these would be individual SNPs that I'd be testing for association. These SNPs are all linked to genes in this particular GO term. This is Golgi-associated vesicle biogenesis. So a bunch of genes related to vesicle biogenesis on this side. On this side, we have a bunch of genes that are SNPs that are linked to genes in the Fc epsilon receptor 1 signaling pathway. So this pathway controls the allergic response, basically. And what we're showing you here are, if I would do a single variant test, these bars reflect the significance of that test. And this dotted line is a marginal 0.05 p-value cutoff. So as most of you know, when you do GWAS, we apply something like a 10 to the minus 8, so something ridiculously high in terms of significance because we're doing so many tests. None of these individual SNPs would get anywhere close to a genome-wide significance cutoff. You can see some of them, if I use a weaker cutoff, I'll get an individual association with the disease. That's not why this is striking. What's striking about this is that we found an abundance of weak interactions between pairs of SNPs crossing these two pathways. So each one of these edges you can see here is a case where when you look at individuals that had a variant in this gene and a variant in this gene, that tend to be associated with in this case, it tend to be associated with the case group. So this is a protective effect. And furthermore, I found many other pairs crossing the same two pathways that had the same effect. So basically, there are a whole bunch of different variant pairs that collectively I can tell there's an enrichment in the case group for individuals that have combinations of those variants. So and you can see some of the stats that supported this. So I expect to get in this SNP-SNP interaction network at the cutoff that we're looking at, I expect to get about 1,500 weak interactions. We got something like 1.5 times that. So there's an abundance of edges crossing this bridge from what we expect by chance. And if I'm testing things at the pathway level, so if I'm looking at about 800 pathways or 1,000 pathways, I'm doing a pairwise test on the pathway. So I've changed the test from single pairs of SNPs to pairs of pathways. That's on the order of a couple 100,000 tests. Now I actually have, I can deal with a multiple testing burden, and a result like this is clearly statistically significant. So this passes multiple test correction of a 0.0. So this is a false discovery. This would be discovered at a false discovery rate of about a 0.02. So we are confident that there's an association between combinations of SNPs where one SNP is in a gene in that pathway and one SNP is in a gene in that pathway. And in this case, that seems to protect individuals from Parkinson's. So I should say that in this particular cohort, we discovered 2 interactions like this at that level. So not a ton of them, but at that 0.05 FDR cutoff, one of those 2 replicated in an independent cohort. So the kind of gold standard in the GWAS world, I can discover things in this cohort. If I can't replicate that in an independent population, a totally non-overlapping set of patients with the same disease, we don't get that excited about. In this case, I can take 2 interactions and I can replicate one of them, which got us really excited about this case. So we're confident in this association being here. I should say, I'm going to skip those details. If I relax that cutoff a bit, so this is at an FDR of 0.25, false discovery rate of 0.25. So 75% of the edges I'm showing you on the slide are real. We can find a bunch of other interactions for Parkinson's. So each one of these nodes reflects a pathway, and the edges are the kind of interaction I just showed you. So for instance, the edge that I just showed you is this edge right here. It's an interaction between the Golgi-associated vesicle biogenesis pathway and Fc epsilon receptor 1 signaling. So that's a protective interaction. Interestingly, you can see that this vesicle biogenesis gene set actually showed up as kind of a hub in this network. So there's a whole bunch of other between pathway interactions that we discovered associated with that. And also there's a within pathway interaction. So combinations of variants within that pathway as well have protective effects in this particular cohort. So at this cutoff, we could find something like 20 interactions, 23 interactions that pass that level of significance. In this case, most of them were protected. This is something that it varies by cohort, varies by disease. This is not something that I would say generalizes that well. We often find increased risk, deleterious effects as well as protective effects. So both things have been discovered by our approach. So that's in Parkinson's case. I'm not going to go into tons of detail about Parkinson's interactions, but I'll just mention. Obviously, the next question is going to be, well, what SNPs, what genes should we focus on? And we are very cautious about doing that. The whole point of this approach is that we can't have much confidence in any individual pair of SNPs crossing those two pathways. We don't have the statistics to really support that. That said, we can kind of look and see which interactions, which SNPs tend to have the most interactions. And I can tell you for this vesicle biogenesis gene set, there are a couple of SNPs and genes that rise to the top of that. For instance, RAB5C is a gene that's involved in vesicle trafficking. And there's a SNP that's involved in a whole bunch of what look to be modifier effects, which gives us a lot of confidence that SNP is worth looking more at. And I should say, I haven't talked a lot about Parkinson's, but there's a whole lot of work in the Parkinson's field about vesicle trafficking. So the SNPs that I showed you mostly don't have single association, but biology in the case of vesicle trafficking is highly associated. This is on the radar of many Parkinson's disease biologists. So the fact that the vesicle trafficking comes up as a strong component of this modifier network is not surprising to people and gives us more confidence that we should pursue that even further. So I had an interesting discussion with the students at lunch. So the question is, what do you do with these kind of results? And there was a discussion about why don't we just apply this across a whole bunch of different diseases. And one thing that we've been working on the last couple of years with these approaches, with these results, is really connecting with disease experts in these areas. So in the case of Parkinson's, we have a collaborator, Vic Kurana, and we've worked a lot on trying to functionalize specific genes that have come out of this and understand whether there's support in models for these diseases for the modifier effects we're having. So what we're finding from the population genetics. So that's a challenging road. We have some exciting results that I'm not going to cover today on that front. Intriguingly involves some modifier effects with P-body genes that came out of our analysis. But that's a story for another day. But I should just mention there's a long road ahead to verifying the significance of these results functionally. So what we have done is applied this to a handful of different diseases. Some of these in collaboration with experts in those areas, others because of interest. So COVID-19 severity, obviously, this is something that many people are interested in the last year or two. And I should say we've in 10 cases we've looked at seriously. So we've got multiple cohorts for most of these 10 diseases we've looked at seriously. 9 of those 10 we've discovered at least a handful, at least one or more interactions. So the Parkinson's case, which we use kind of as our test case, definitely is an interesting case, but we can find genetic interactions, evidence for genetic interactions with a lot of these different diseases. So Parkinson's, schizophrenia has actually been fairly productive, a number of other diseases as well. Also, cancer susceptibility is something that we've worked quite hard on recently. And I will say COVID-19 severity. So these cohorts are increasing in size. So the UK Biobank now has a pretty decent chunk of patients that had COVID-19. And what we've done is done an association test with the severity of the COVID. So all of these are COVID-infected patients, but we've looked for interactions that determine the severity of outcomes in that case. And we found some interesting things there involving HLA types, and I'd be happy to talk offline if people are interested in that. If I look across all of these cases, about 25% of the pathways you identify you get by traditional approaches. So if I just do a single variant test and do some kind of a pathway enrichment analysis on that, about 25% of those cases you would find. So a lot of the pathways we're finding you can only get if you start to take a modifier view of this problem. So we're excited about that. We're anxious to explore many of these results in these several of these diseases in more depth. I'm going to skip over that. So I want to transition here for the last couple of minutes of the talk So ongoing efforts. So as I said, we're kind of diving in detail. We have a major effort in Parkinson's, Alzheimer's, and cancer right now. So with funding from the Michael J. Fox Foundation and Alzheimer's Association, we've really done a deep dive on Parkinson's and Alzheimer's and trying to understand if there's a common basis if we start to look at modifiers there. We have an ongoing project in understanding genetic interactions across environments. I mentioned that briefly in diverse genetic backgrounds. So that's something that we continue to be interested in. Yeast is a great model for that still. I want to talk a little bit about another emerging project for the last couple of minutes, which is actually going to human cells and trying to map interactions in the same precise way that we did in yeast, and trying to understand genetic interactions that way. So obviously we can try to define interactions associated with disease from populations. The much cleaner way to address this problem is to go into human cells and edit them in combinatorial ways and measure phenotypes, just like we did in yeast, and approach that problem that way. So I'll give you kind of a preview of how things are working. This is a project that's been ongoing for several years in the lab, but it's now coming to fruition. We're about ready to publish the first kind of big human genetic interaction map that was produced in a similar fashion to what we did in yeast. So I'll try to give you a preview for what's happening on that front. So the basic approach, and again, this is a collaboration with the Boone and Andrews labs. We've added a collaborator now, Jason Moffett, who has a lot of expertise in human functional genomics, setting up large-scale CRISPR screening approaches. Try to sketch out the approach we're taking. So we've committed to a single human cell line called HAP1 cells. It's a CML-derived cell line that was forced to be haploid. So we thought that was a great system to do some kind of genetic screens in. Turns out that cell line likes to diploidize a lot. So, and that doesn't really pose a problem for us in these screens anyway, but that was the original motivation for picking HAP1 cells. We view this as a model cell type. We want to map a comprehensive network in that cell type, much like we did in yeast, and we view this kind of as a reference map. So that's the goal in that context. And the way we're approaching this problem is like this. So we take HAP1 cells, we make a specific query mutation. So we pick a gene A, say, we knock out that gene using kind of directed CRISPR approaches, make a stable cell line that's got a loss of function mutation. We can verify that through sequencing. And then we do a genome-wide genetic screen enabled by CRISPR in that background. So basically, you make a single mutant and then do a screen targeting about 17,000 or 18,000 genes, each one with about 4 different guides. So this is in a pooled setting, growing cells on plates. So each one of those screens then basically fills in a row of our matrix. Think of it like a single genome-wide screen that gives us quantitative interactions for this gene A with all other genes in the human genome. And then we repeat that process. So we pick a different gene, let's say gene B, make a loss of function mutant in that, do this genome-wide screen, find positive or negative interactions in much the same way that we did in yeast. So that's the approach. This is not the only approach you can take. There are other people that are taking, let's say we're kind of filling in one row at a time of this matrix, but each row is complete. You can imagine taking, say, a group of about 200 genes and doing all pairs in that 200 by 200 space. So Jonathan Weissman's lab has done some nice work there. We're taking kind of a genome-wide approach. We want to try to get a profile for every gene in the human genome, even if it's across a small set of genes that we pick. So we're trying to be smart, use our yeast expertise about how you pick genes to best light up this network. So for example, to return to some discussion we had earlier, we're picking genes that have subtle phenotypes on their own as a basis for these query mutants, as well as spreading those mutants across function as well as we can. So we're trying to light up the human functional map as well as we can. So just to show you what the data from one of those screens looks like, this is a gene called PTAR1, which has a role in secretion. So this was a genome-wide screen on a loss of function mutation in PTAR1. And what I'm plotting here is the fitness. This is basically the log fold change in HAP1 wild type cells versus what happens when you knock out PTAR1 for those same genes. So genes over here are genes that had much stronger fitness defects. Blue here is for negative interactions. Yellow here is for positive interactions. These genes down here had much stronger fitness defects in the background of PTAR1 knockouts. This is a genetic interaction basically. So these things are tending towards synthetic sick or synthetic lethal. And you can see RAB14, this is a secretion related gene, a lot of vesicle transport, Golgi transport related genes, which makes a lot of sense for PTAR1's function. So on the positive side, I'm pointing out one case here. So PTAR1, so one of the positive controls you get in these screens, if you knock out the gene in the library, when you target the gene that was already knocked out in the query cell line, you should expect no additional fitness defect, and that's what we see in this case. So PTAR1 basically, it has a fitness defect in wild type cells, but as soon as you put you do a screen in a PTAR1 knockout, you get what looks like a positive. This is kind of a technical control positive interaction. So in general, these screens work. So you get genes that make sense. I'll give you show you some results in a second that we can kind of play some of the same games we played in yeast from these. But in general, we're after several years of kind of working through technical details of these screens, we think that we can get quantitative genetic interaction profiles for a fitness phenotype in this model cell line quite well. So to date, just to give you an update of where we're at with this mapping project, again, this is 5 or 6 years of work in this human front. We have something like 200 query mutants completed at this point. So each one of those 200 mutants was crossed into about 17 or 18,000 genes. So after quality control, we have about 17,000 of those genes left. So this is a very skinny, long matrix, right? So we don't have a ton of the human genome queried, but most of the genes in the genome appear on this side of that matrix. And that means I can start to play some of the profile similarity games that we played in yeast. So for example, I can look at what is a profile of interactions that a given human gene has against these 200 mutants, and I can start to localize on it functionally based on just how it responds against these well-chosen, hopefully well-chosen 200 query mutants. So that's something like 3.4 million gene pairs that we've constructed so far in human cells. That crank is still turning as we continue to optimize the approach. But we're now ready to publish kind of the first reference map that's got reasonable genome-wide coverage. Of course, we're covering a tiny fraction of all possible gene pairs in humans. So I have a couple of other kind of data slides. Like this might not be very easily to view especially from the back of the room, but the point is, if I zoom in on that clustergram that I just showed you, we get very encouraging results. You can see a whole bunch of Golgi associated genes clustering well here. I talked with a few of you about mitochondrial genes in these genetic screens. This is a superstar of really any of the human CRISPR-based screens, as far as we can tell. We can recover the whole, a whole bunch of mitochondrial biology quite well. The whole respiratory chain complex comes up. Mitochondrial ribosome clusters quite well on these screens. And in general, that's what the profile similarity network looks like for humans. We can put about 5,000 genes. So there's about 10,000 expressed in HAP1 cells. We can put about 5,000 on a fairly confident functional map based on this profile similarity approach. And using the same kind of tools that we developed in yeast, many of those neighborhoods that you can see clustering are strongly enriched for biology that we know about in human cells. So now we've got kind of our unbiased functional map for human cells that we were after, and we can start to really dissect the biology that's being highlighted by these genetic interaction clusters derived from genetic interactions. So that's a very brief overview about what's going on that front. So I just want to kind of summarize things. So I covered a lot of ground today. Hopefully you have a sense. We've finished a comprehensive map, genetic interaction map in yeast. We learned a lot about principles about genetic interaction structure. I think a lot of those principles we can translate to study human disease. One of the successes of that in the last couple of years have been to develop that leverages the structure we expect, and that reduces the multiple testing burden. We can now start to systematically discover pathway level genetic interactions from human population data. And we think that there are many diseases where those interactions are relevant. We're finding evidence for those interactions. And we're starting to map some of these experimental approaches to human cells, as I mentioned toward the end, in terms of the reverse genetic approaches to mapping. So I want to acknowledge two specific people for the bridge work, the sort of main focus of the talk is this bridge method. So Wen, who was a PhD student in my lab, is now senior research scientist in the lab, and Gang Feng, who is a student in Vipin Kumar's lab at the University of Minnesota. He's now a professor at Mount Sinai School of Medicine in New York. So they really drove the development of that bridge method I mentioned. Everything I do is a huge collaboration with a lot of people. I should really acknowledge in addition to Vipin Kumar's lab at Minnesota with the bridge work, the Anders and Boone labs at the University of Toronto, everything I've done in yeast is really a joint work with them. Jason Moffat's lab for some of the human CRISPR screen work that I mentioned towards the end. Funding from NIH and NSF over the last several years. And I'd be happy to take more questions if you have them.
Great seminar. I have questions.
Hey guys, really enjoyed the talk. I had a question about, you mentioned there are a bunch of genes that come up in the yeast that don't show interactions. Have you considered doing three-way interactions and guessing at redundancy to try and reduce the multiple testing?
Yeah, it's a great question. So we did do a survey of triple mutant interactions. So we published that in 2018. Again, we're scratching the surface on that. That space is huge. I think we constructed a couple 100,000, maybe 300,000 or something. So it was less than 1 million, but several 100,000 triple mutants. There are motifs in the double mutant network that are predictive of the presence of triple mutant interactions. I think this idea, though, that we somehow, at some point, we're going to discover phenotypes for these genes that are dark. Again, that didn't work out that well in that case. The things that dominated the triple mutant network also had a lot of single and double mutant phenotypes. So, yeah, the dark net's been elusive no matter how we slice this problem, I would say. There's interesting exceptions to that. I think genes with small family sizes, as soon as you cut through that redundancy, you'll start to see phenotypes. So I think there's interesting exceptions to that. But I would check. There was a paper from 2018, Kuzmin et al., Kuzmin, if you're interested in the triple mutant space. It's a good question.
Last up. So that was a great talk. I had a couple of questions. One, maybe you mentioned it. How much, well, I'll ask both my questions. How much more of the heritability can you explain in family interactions?
Yeah.
And the other question is, I'm curious how you envision using the data from your HAP1 interactions. Now, if you return to the GWAS analysis, how can you use that data to look for real variance in the population? Yeah,
those are both great questions. I'm going to take the second one first. So this is a really productive line of research, I think. So the really great thing about the HAP1 genetic screens is we're making perturbations on genes. You could call this a weird cell type. Who knows how relevant it is to any given disease? But what it tells us is if you perturb that gene, and pretty strongly, we're making basically loss of function mutations for the most part, you will get a phenotype, and that's really valuable information, right? We know that you can make a single perturbation or double perturbation and see a phenotype. And furthermore, the clusters that I showed you here, these are perfect clusters to seed an approach like bridge. These are clusters that aren't just annotation based or knowledge based. These are clusters that phenocopy, genes phenocopy each other, at least in this one human cell type. So one of the things we're doing is taking these clusters and using them as a seed and input for the bridge approach. And the kind of things we talked about with the peroxisome, I think any place you want a related set of genes that's likely to cause a similar phenotype, these kind of networks I think are great for that, these genetic based. So there's a lot of applications for HAP1. As we get other cell types, I think that's going to be a really synergistic interaction in a research sense. The other question about missing heritability, how much do we explain? So this is really unsatisfying. So I think anyone that's worked on human genetics data, when you discover an effect in a data set, we overestimate its true effect size, right? Because this is the sort of so-called winner's curse. You find it because it's really strong. That is almost never that strong if you look in another cohort. So what that means is it's really hard to estimate so the true impact on how much. So in the data sets we discover these things, we can explain a ton of heritability. Does that mean that we do in reality explain a bunch. That means in that discovery data set, I can explain a bunch of the case control relationships. So it's bottom line is it's hard to get a true estimate on how much more we are to explain. One thing we're working on that's more concrete than that is how much do predictive models, like PRS models, polygenic risk scores, are really common. Can I predict disease status or subtype based on a usually linear model approach? If I use these interactions in those models, can I get better discriminative power? And that's I think maybe the more concrete way to prove or demonstrate the additional information that these interactions provide. So sorry, it's kind of a fuzzy answer to the question I used to motivate my talk. Yeah.
No, you can talk more about it.
Yeah.
I think it's interesting and might be telling about the genetics, but how you explain the.
And I think estimating heritability on data sorts where you discovered loci is a huge elusive challenge. I wish there were a better answer to that.
Right. So when you're looking at the interactivity between two pathways, do you expect that like the individual genes or the individual components of these two pathways have the same kind of interaction? So let's say that like between pathway A and pathway B, individual components could have like both negative and positive effects. I guess it sort of depends on how you define the pathway, right?
Yeah.
And does that sort of like make it difficult for anything to stand out because these effects could sort of be canceling each other out?
Yeah, that's a great question. I think in yeast, the answer is simpler because we have, in many cases, we have clean loss of function or we have highly deleterious hypomorphs. I didn't talk about the essential genes at all, but essential genes, we have some hypomorphic alleles. We can study interaction for them, but there it's very rare to see mixed bridges. The data is depleted for bridges that have a mixed interaction type. That is, when you find one pair with a negative effect, almost all of them have a negative effect. The answer is, I'm sure not that clean with real human variants. And there's good examples. In GWAS, you can get one variant in a gene has a positive protective effect. The other one has a risk associated effect. So it's way messier in sort of the natural variation in human populations. So we're kind of simplifying. We're looking for things with coherent effects when we discover them with bridge. We're missing a ton of interesting things like what you're suggesting. I'm sure they exist all over the place. That if I can find a deleterious effect between these pathways, I'd be surprised if there's not some other combination of variants that the phenotype goes the other way. I think that's fairly common in the natural variation space. There's a disconnect between natural variants and populations and our contrived knockouts in yeast, and that's something it's an interesting problem to grapple with, I think.
Yeah, I was just wondering whether you could use your system to study known disease risk genes. So like RAS-BEL12 or BRCA1 or 2 or SOD this, and look for modifiers where an individual may be a carrier, but it doesn't show the disease because of the background.
Yeah, it's a great question, and we have done this for a few cases. I think it's a really productive because that constrains our hypothesis space even more, right? So, and in fact, this is what I mentioned, this Peabody study that we have coming out. This is with Vic Khurana's lab. So in that case, we showed that variants in alpha-synuclein, which is associated with a variety of diseases, neurodegenerative diseases, are often modified by variants in Peabody genes. And this is something that, of course, we knew about alpha-synuclein. We seeded the whole approach with that and looked for individual genes with this interaction approach. So you can do more specific hypothesis tests because you're constraining the problem when you approach that. And I think that's very productive. So if you have a disease that you're interested in, that you have candidate genes you want to look or variants that you want to look for modifiers, we'd be happy to work on that with you. And I think it's a very productive direction.
Are there any more questions in the chat?
Thanks a lot.